we are at the Michaelis Institute, a mixed research unit between INRAE, AgroParisTech and University Paris-Saclay, located at Jouy-en-Josas near Versailles. Our aim is to decipher the interaction between food, foodborne microorganisms and the host to explore the link with health and disease associated with pathogens. When possible, we translate our research to societal application in the field of food safety, health, biotechnology and food fermentation. We host the Ferment du Futur distributed PIAM platform that is devoted to the study of microbial interaction with food matrix for industrial and health application.
Ferment du Futur is a public-private partnership that aims at accelerating innovation in the food sector by harnessing the power of ferments and fermentation. The investment also allows for the purchase of state-of-the-art technologies that are set up in existing research units with world-leading scientists such as Michaelis. The new MICA microscope was selected for its powerful capacity to precisely elucidate how microbial communities interact with three-dimensional food matrix and profoundly change their chemistry and nutritional properties.
The MICA wide-focal microscope is integrated within the MIMA2 imaging core facility here at Institut Michaelis. The MICA is installed in this L2 biosecurity lab. So what we like about the MICA wide-focal microscope is that it's pretty user-friendly. Users can get to do imaging pretty fast. In about 10 minutes, you explore your sample. This allows users to be able to explore the three-dimensional sample, so for doing Z-stack imaging or time lapses in order to understand the dynamics of the sample. This microscope is well adapted to explore live food matrices such as the kefir three-dimensional biofilm. We can also use for studying host pathogen interactions as Salmonella biofilm formation. The MICA microscope is also well adapted to explore in more higher resolution interactions between pathogens and cells within the cell, as for instance the invasion of Listeria monocytogenes. MICA is well adapted to incubation situations that are more delicate for a microscope, such as temperature that can go until 40-45 degrees. So this gives us a full panel of diversity of samples that we can explore with the microscope.
The wide-field MICA technology can assist us in three directions in our field of research. The first one is high throughput. We need to get high throughput information on spatial structure to be able to relate those spatial organization data to genomic information to identify genes involved in the emergent properties of this community. The second point is interaction. We need to study interaction at high throughput, whether they are cooperative and beneficial in the community or beneficial pathogen interaction mechanism. And the last point is spatial transcriptomic. Using this technology, we are now able to follow the expression of a single gene in a spatial population, taking advantage of fluorescence reporter genetic construction. It is clear that fluorescence microscopy will make a difference in the coming years in our field of research to understand the emerging properties of those spatially organized communities that we find in food matrix and biofilm.